So in Unit 2, we're going to begin our study of cells. In Section 2.1, we're going to focus on cell structures. So what are the different types of cells, what parts make those cells up, and what do those little parts do to help the cell function and overall help the organism function. So in Section 2.1, we're actually going to be on page 23 of your notes. So if you'll turn to page 23. Before we start, this is a picture of a bovine pulmonary artery endothelial cell. And really what this means is this is actually a sheep cell, and this has actually been fluoresced. There's a chemical that's been injected on it, or that it's been stained with, that's helping it shine with those vibrant colors. And so we're really going to focus today just on what are the basics of the different types of cells. And then tomorrow, or for our next lecture, we're going to talk about what's actually inside of the cells. So notice that there are different colors, and it seems like there's different parts there. And that's actually because the stains that are used will attach to the different types of tissues or the different types of organelles that are there. So talking about cell structure and function in this section 2.1, we're first going to remember that macromolecules such as proteins, carbs, lipids, and nucleic acids are organized to form these structures that create the cell. Remember that we have our atoms form these larger molecules, and then the cell itself is made up of those molecules that eventually forms your living thing, your living organism. Here we have another example of some of those tissues or some of those cells. This is actually a cross-section of skin cell or skin sample. And so at each layer here that you see, that's actually a different type of cell. On the very top layer of your skin, those cells have to be able to shed. They have to be able to wear away and grow back pretty rapidly as well. And as you move down, you have more complex cells, cells that secrete oils on top of your skin, and then cells that help you feel the things that you touch. One of the first scientists that we have to talk about to learn a little history about the cell is Robert Hooke. Robert Hooke was actually the first to observe cells, and he actually observed cork cells, which we're going to learn are a type of plant cell. So he looked at cork cells. This is a type of plant cell. And all he was really able to see, because he had a very primitive microscope, is the little outline of the cells itself. He wasn't able to see anything inside of the cell, but he was able to see those individual shapes. So just think of the cork on top of a vinegar bottle or on top of your parents' wine bottle. Okay? It was very similar to that, and that's what he was first able to observe. Do notice that these are dead cells. He didn't observe the living cells yet. He didn't really know what they are. But he was a priest or a monk, and he lived in a little room that was called a cell. And so these little spaces or these little shapes that he saw reminded him of those rooms, and so that's why he named it the cell. The second important scientist is Anton van Leeuwenhoek, and he was the first to actually observe our living cells. And he actually looked at some cheek cells. Now later on we will also be observing our own cheek cells, but notice that they're very faint in color, and we're actually going to use a dye to be able to see those cells a little better. All he was able to see was kind of the exterior of that cell, and a little black dot at the center that we'll talk about a little later on, what that was. But he was the first to observe those living cells. Now there's four main structures that we're going to talk about that are found in all cells, okay? The first is the cell membrane, and this is a phospholipid bilayer. So here are our lipids, our fats that we've talked about when we talked about macromolecules, an organic macromolecule that's made up of bilayer. So there's actually two layers of those phospholipids, And so remember that our phospholipids kind of looked like this. There was that phosphate head and those fatty acid tails, and now there's actually 2 layers of them. Okay. And they actually turn in on themselves, so those fatty acid tails are away from water. That water could be water outside of the cell, and that water could be water that's in the cytoplasm, or the liquid inside of the cell. But either way, you have these 2 layers of phospholipids that are surrounding the cell. So here are 2 pictures of 2 different types of cells, a prokaryotic and eukaryotic cell, and we'll talk about what that means in a second. But on each of them, they have that cell membrane on the outside. Sometimes it's also called a phospholipid bilayer. Sometimes it's also called a fluid mosaic model, because there's lots of different parts inside of it. But the next part is the cytoplasm, and that's the filling, or the inside filling of the cell. And it's made of mostly water. So all of the different parts that are inside the cell, the different organs or tiny organelles that are inside of the cell, will be floating inside of that watery filling, okay? So all inside here, it's not an empty sac, okay? The membrane's not an empty bag, but it's full of water and other parts of the cell. Then the third structure that all cells have, prokaryotic and eukaryotic, is DNA, that genetic material to control the cell. Remember that DNA was one of our types of nucleic acids. Okay. They're organized into what are known as chromosomes that are circular in prokaryotes and linear in eukaryotes. So in a prokaryotic cell, if you notice in this picture, the DNA is like a little squiggle inside of it, a little loop. But then when you look at the eukaryotic cell, the DNA inside here will actually be a little more straight, okay? It might look like that double helix that we've seen before, or it might look like a little X for a chromosome, and we'll talk about that more later on as well. Then we have our ribosomes, and ribosomes are an organelle where DNA code is used to build or synthesize proteins. So notice that we have all 3 of our 4 organic macromolecules. We have the lipids in the cell membrane. Remember that there's some carbohydrates in there as well. In the cytoplasm, we have water, a lot of inorganic material. We have DNA, which is organic, and then we have our proteins being made. I told you that we have all 4 of those macromolecules inside of every living thing. And so right here, we see 3 of them already, and we'll talk a little more about carbs later on and how they're used inside the cell. Now the types of cells that we have, there's 2 major types of cells, and that's going to be your prokaryotic cells and your eukaryotic cells. Now pro actually means before, so we're trying to break down this word, and kary means nucleus, okay? So pro, before the nucleus, prokaryotic. They do not have a central place to store DNA. So a nucleus is a central place to store DNA. Do not have a place to store DNA. Really important for you to remember. The DNA is free floating in the cytoplasm, so it's in that liquid. And these cells have all 4 main organelles to build a cell, but are able to perform all Stirninger-like functions. So they only have those 4 main parts. They have a cell membrane, they have that DNA, and it's usually in the form of a loop. And we have the cytoplasm, and we also have those tiny ribosomes. We'll see later on what they look like. Tiny ribosomes to make proteins so that they can function. They will perform all of Stirninger. One thing to keep in mind for our prokaryotic cells is that they are very simple cells. They are simple, and the examples we have, or the kingdoms of life that are part of that prokaryotic cell, is our archaea bacteria and our eubacteria. So we've mentioned those kingdoms in our last unit. And the two kingdoms that belong or are made up of prokaryotic cells are those two. Now the last type of cell we have are the eukaryotic cells. EU means true, okay? And karya again means nucleus. So these are the cells that have a true nucleus or a true central place where their DNA is stored. So the DNA is just not floating around there in the water, in the cytoplasm, but it has like safe place to put it in. Now these cells have a membrane that surrounds all the DNA in the cell, so that's the nucleus. And the cells have all 4 main organelles, as well as up to 7 additional organelles to perform specific functions. So we will talk about what those functions are later. But as a little reminder, for eukaryotic cells, there's several kingdoms that belong to, or that are made up of eukaryotic cells. That's going to be our plant cells, our animal cells, our fungus, and our protists. So we had 6 kingdoms, and these 4 kingdoms are made up of those eukaryotic cells. They're more complex. They have a true nucleus. They have a true place to hold their DNA. Okay? And then the last little thing at the bottom of your page, you have a little summary box. I want you to go back to your pink vocab sheets. Try to see if you can use 1 word per sentence, and I actually want you to create 2 sentences that summarize this section of notes. So try to use at least 1 vocabulary word in each sentence, and try to make connections or comparisons between the prokaryotes and the eukaryotes.